Great. All right. Well, welcome everybody. And thank you for coming today. My name is Darcy Dugan, and I'm with the Alaska Ocean Observing System and the Alaska Ocean Acidification Network. Excuse me. And I'm joining today from the traditional land of the Dena'ina people where I live in Anchorage. And I want to introduce session 2 of our 4-part series hosted by the Alaska Ocean Acidification Network. And the OA network started in 2016 with the mission of trying to expand our understanding of ocean acidification, both the processes and the consequences, as well as looking into adaptation and mitigation opportunities. And today's discussion falls under one of the primary objectives of the network, which is to bring together experts on what we know so far about ocean acidification in Alaska, and also people to whom those impacts most matter. So these next 2 sessions are on species response. And today we'll be looking at mariculture and subsistence species. And then on April 20th, 2 weeks from now, we'll be looking at commercial species. And today is building on session 1 in late March, which was focusing on regional conditions and what we know about the waters around Alaska. So similar to last time, we're going to start off with 3 speakers. Today we'll be hearing about Pacific oysters, which are the primary shellfish farmed in Alaska, and then subsistence species, including clams, Darky, and mussels. And then we'll be learning about how tribes and communities have been involved. And then we'll have a chance to get into discussion in breakout sessions. And this will be an opportunity to ask questions about what we heard during the presentation, and also think about ways that we can further collaborate or communicate this information. So we're looking forward to a good multifaceted discussion. I want to also thank the executive committee again of the Ocean Acidification Network, because they've put a lot of thought into kind of setting this up and how to structure these discussion series sessions. So I think Molly is going to put the names of the executive committee in the chat so you can see them. So we want to extend our gratitude towards those folks. And then I will introduce Molly Mayo, who is our facilitator today. She's with the Meridian Institute and has been focusing on natural resource management and policy issues through facilitation and mediation for the past 25 years. And she's going to outline how today will work. But before she does, I'm going to put a quick poll question in the Zoom. So if you go down to the bottom of your screen and click on poll, which I will activate right now. Let's see, launch. This will help us get a sense of our group today. And now I'll hand it off to Molly.
Thanks, Darcy, and welcome everyone. I'm really excited to see the turnout growing here as we continue and have a terrific set of speakers that I have the honor of introducing to you today. While we see a couple more people joining, let me just say that in an effort to make sure that we manage the noise, please try to keep your audio muted. It looks like everyone has already done that, but we'll keep an ear open for that. If you could just make sure you stay muted. We're going to use the chat function to allow some interaction during the full plenary, but since there are so many of us, we'll rely on the breakout sessions for more active participation from everyone together. So for the plenary session, feel free to share questions or comments in the chat. We'll be tracking those, and where appropriate or where there's an opportunity, we will try to create space to address if there is time. In the meantime, if you would like to, you're welcome to rename yourself with your organizational affiliation. I see everyone is already pretty well named with your common name, but feel free to add your organizational affiliation if you would like to introduce yourself that way to the full group. And then rest assured, we'll have a little bit of time in the breakout session for you to introduce yourselves in another way. So we also take advantage of the opportunity to get to know some new people here. So as Darcy was explaining, our goal is to not only hear from experts, but also generate some discussion, because all of you are experts in your own way. And this is the latest in a series of 4 different sessions where we are going to dig more into what we understand about ocean acidification and the impacts that it's having in Alaska. So we really want all of you to be thinking about how you can add your voices to what we understand. So please be prepared to actively participate during the breakout session when we have that time. So for now, I'll move into introducing the first of three speakers, as Darcy said. What we're trying to do is to better understand what we do know about the impacts of ocean acidification on mariculture and subsistence species and the communities that depend on them, and also to understand what we don't know and what we're trying to better understand more about them in the future. So our first speaker is Iria Gimenez, who is a biological oceanographer and marine ecologist and a postdoctoral research fellow at the University of British Columbia and the Hakai Institute. Her current focus of her research is on the biological impacts of ocean acidification, particularly on shellfish and invertebrates as a whole. She's developing new experimental tools and approaches to better replicate the complex natural conditions through laboratory experiments. So Iria, thanks for kicking things off for us, and feel free to share your slides.
Thank you for that introduction, Molly. Just making sure that you are seeing my slides correctly.
Looks good.
Yeah.
Okay. Well, thank you everybody for attending this workshop, and thank you Darcy and Molly and the Ocean Acidification Network for inviting me. I have to say, first off, I am working currently in British Columbia, and some of the work and thoughts that I'm going to share today are also coming from previous work that I was involved in Oregon and Washington State. Unfortunately, I don't have a lot of experience with shellfish research in Alaska, but I'm hoping that during the breakout session, we can discuss a lot more of that. So the talk today is going to be very focused on Pacific oyster, but throughout the talk, there will be some times that I'm going to talk a little bit more about things that we've learned from other oyster species, and in some cases from other bivalve species that kind of inform the story that we're putting together for Pacific oysters. So there we go. So what does ocean acidification mean for the Pacific oyster? We know that they're vulnerable, and they display negative responses on a wide range of processes across molecular, physiological, organismal, behavioral, and also ecological. So at multiple scales of organization, multiple types of processes. But we also know that throughout the life cycle of a Pacific oyster, not all stages are equally vulnerable. There are certain stages that offer or display heightened vulnerability, and we usually call those these windows of vulnerability. Sometimes we also observe that impacts that are experienced during these vulnerable stages sometimes manifest themselves later, and sometimes they also carry over to a different life stage. So that's not to say that when an oyster larvae is impacted by acidification, that's the only time that that impact is going to be suffered.
It's just boring.
Okay, so we're gonna delve a little bit more into that. I'm gonna try to give an overview of all we know in all these different life stages throughout the life of an oyster. But I'm gonna be using this schematic that you see in the screen across the talk, and I kind of wanna briefly walk you through it. So if we start, I think you can see now my point, right? Yeah. So if we start here, Pacific oysters are broadcast spawners, so they release the sperm and egg into the water, and they get fertilized, and that starts their larval period. So these early larval stage, we already know is a very vulnerable period for ocean acidification impacts. Then the larvae keeps growing. In this later life stage, is less vulnerable. So I've marked it as blue, but that doesn't necessarily mean that they're completely immune at this stage. Then towards the end of their larval period, they go undergo metamorphosis. They find a suitable place to settle, and they metamorphose. These metamorphosis and early post-metamorphosis stage is also a very sensitive stage and sometimes considered a sensitivity bottleneck. And then they grow into adults, completing the cycle. This period of adults is not necessarily as vulnerable, but we'll talk about some of the other effects that we do observe during this time that sometimes can carry over to the next generation. Sometimes, and I want to bring this up early so we can kind of keep thinking about it throughout my talk. Sometimes these kind of rapidly shifting environmental conditions that we're observing, particularly across the waters of the Pacific Northwest and all the way into BC, do happen fast enough that natural selection might not be able to keep up. So this is something that is important to keep in mind when we're thinking about potential mitigation strategies. But we do see differential responses in different populations. So there is the capacity for some level of genetic adaptation, not necessarily will be the solution to all the problems. Because of this, there's also a different kind of process that we term transgenerational plasticity, which is not necessarily operating in the same scales as natural selection, but it really is parents given a memory of what an exposure to acidification meant and passing that memory to their offspring. This process operates much faster, and we're starting to learn that maybe this would be a very important process to keep in mind when we're thinking about rapid adaptation to these environmental changes. Okay, so I have to start here because a lot of what we know about Pacific oyster and acidification really was kickstarted by the problems that the shellfish industry started having in Oregon and Washington in the early 2000s. So I'm sure many of you here know the story, but briefly, Whiskey Creek Shellfish Hatchery is located in Oregon, and it's producing seed for most of the independent growers from California, actually from Mexico all the way to Alaska. And in 2007, a little bit earlier, depending on who you ask, they started having tremendous production failures. Researchers from Alaska State got involved, and they discovered, alongside with the hatchery operators, that exposure to corrosive conditions in the beginning of the larval life actually impacted the production in such a massive way that just by looking at those first 2 days of the larval life, they were able to explain most of the problems that they were having in the entire cohort, entire larval production. They particularly narrow it down to saturation state. Saturation state is a measure of how corrosive the waters are, and in other words, is how easy it is for an organism that makes a shell to build that shell. So when saturation state is low, towards 1, that means that dissolution of that shell is favorable. And when it's above 1, it means that keeping that shell from dissolving is the process that is favored. That is chemical thermodynamics. That doesn't necessarily always mean that 1 is the magic number. In fact, for oyster larvae, 1 is not quite good enough. Conditions have to be closer to 1.5 to 1.6 saturation state for them to really be able to build that shell comfortably. Because below that number, even if keeping that shell is still favorable, it takes a lot more energy. So they've identified this window of the first 2 days, but why? So that's where part of my work comes in. We did experiments in the lab where we manipulated that saturation state, that corrosivity of the water. And what we found was that when saturation state, again, was a little bit over 1.5, the larvae developed fine. So they didn't look abnormal, and they also were bigger. So to give you a visual of how an abnormal larvae looks like, These are other larvae that we grew from Washington State. And on the left, you see larvae at different stages, day 1, day 2, day 4, that were grown at a aragonite of 1.6 or higher. And then on the right, you're seeing larvae of the same ages that are grown when saturation state was well below 1.45 at this in this case. And larvae were smaller, but also you can see that there's obvious deformities in the shape of their shell. These abnormalities in development usually lead to death a little bit later in their larval life, which is what the hatchery operators were observing. So we know what, but we don't really know why yet. And why sometimes is important, because if we don't understand the process, it's really hard to find ways to solve it, or even to understand how that might affect them later. So we drilled a little bit deeper into what was happening, and we followed larvae very closely while they were developing. And we realized that really the problem for Pacific larvae is that they're hit by 3 different things at the same time. When they're building that first shell, in the first 24 to 48 hours of their life, they build a shell basically in 6 hours. So here you're seeing pictures of oyster larvae at 10 hours after they're born. They have no shell. And then 6 hours later, they have built a perfectly big, covering all their body shell, which means that they've gone from 0 calcium carbonate in their body to 90% of their weight being calcium carbonate in a matter of 6 hours. It's one of the highest rates of calcification in the world during this particular stage. But they're doing that while they still cannot really protect themselves from the surrounding water. That's where having a shell comes in. So if the conditions out around them are not great, they are very exposed at this stage, way more exposed than they will be later on their life. And also they have very limited energy. And that is because they're not feeding until they're building this first shell. They needed to actually be able to anchor their feeding organs. So at this point, they're completely relying exclusively in the eggs that they had, in the reserves of energy that came from the egg that they originated from. So we put together this kind of conceptual model. Why are they sensitive? And they're sensitive because during these states, again, they have a very high calcification rate, but then it goes down as they become older larvae. They have dwindling reserves of energy in the form of lipids, and then they have a higher contribution of inorganic carbon, which is contribution from the surrounding water in making that shell. Those 3 things change throughout the life of a larvae, but they become, again, a little bit more sensitive towards the end of their larval life, because they have depleted a little bit their energy reserves, and they're starting to build them up again, but they're gonna stop feeding again because they don't feed during metamorphosis either. So larvae are very vulnerable. Like I mentioned before, later larval stages are also not completely immune to acidification effects. If you recall that first schematic, we're here now, we're blue, which usually means sublethal effects. So exposure to acidification doesn't necessarily kill you at this stage, but it does leave you with a mark that then sometimes can keep, like you can keep it and take it with you to the next stage, and might be the difference between surviving or not making it. I'm showing you here results from a study that I carry in Whiskey Creek, actually, in 2018. And what I did was I raised different families of larvae from embryos to the end to pre-settlement. So right when they were getting ready to metamorphose. And some of them, I raised them in buffer water, which is what the hatcheries did. They increased the saturation state of the water artificially to make the water better for the larvae. But then I also raised some larvae in totally treated water as it was coming in from the bay. And after 2 days, I changed them, and some that were in buffer water went to unbuffered water, and all the other possible combinations. What I found is that, as I was expecting, larvae that were buffered during the first 2 days of their life usually did better in disease survival until the end. But I also found that even larvae that were buffered during the beginning, but then changed to unbuffered water, generally did worse. So there was an effect of that exposure later in life, even if way smaller than obviously this very early stage that is very, very vulnerable. You're seeing here also results from another study by Co and co-authors in 2015, and they also found similar results. They were looking at metamorphosis at in this case, and they found the same thing, like larvae that were exposed to lower pH, which is here marked with a P, or combinations of temperature and salinity and low pH. They usually found that exposure to low pH was the most important factor here, and it delayed metamorphosis. So like I mentioned before, then we move into the juvenile stage, and this is another critical point where metamorphosis is delayed when the larvae are exposed to low pH. As you can see here, this again, P is low pH and temperature, or temperature and pH at the same time. Then we also had other studies done before on the native oyster in California, where Annelies Hedinger and some co-authors found that when they expose the the native oyster larvae to pH and then grow them until juveniles, the juveniles also carry over that effect and grew less, which is what you're seeing here. These larvae were grown in low pH and kept at low pH as juveniles, and they kept showing a net growth rate for juveniles. So even if this stage has not been well studied in Pacific oyster, there is evidence coming from other species that this is also a potential bottleneck for this organism. Adults, like I mentioned before, are usually more resilient. So we're not necessarily seeing tremendous mortalities associated with corrosive events. But we are seeing reduced growth, and we're seeing a host of sublethal effects. And by sublethal effects, I mean exposure to corrosive conditions that don't kill the organism, but leave a mark in some physiological or behavioral response. There are many studies out there that measure different things, and I've tried to summarize here. So on the top panel, you're seeing as increased PCO2, there's a reduction in the gasification rate of adult oysters. This was a particularly short study, but it's been replicated since in other species of oysters. They don't necessarily stop growing completely, they just grow slower. There's also studies that show increased feeding rates to kind of match increased energetic costs when they're exposed to acidification, and also increased gaping activity. So that's what oysters do when they're particularly stressed. At a molecular level, many researchers have documented increased cellular stress and oxidative damage. That is particularly important because it's very related to immune status. So a lot of what other researchers have found is that when adult oysters are exposed to corrosive conditions for long enough, they become more vulnerable to disease. And they also become more vulnerable to exposure to heavy metals, toxins from harmful algal blooms, and potentially other pollutants in the water. Here you're seeing work from Ko et al. That measured a whole bunch of cellular responses that were usually elevated under lower pH. And then they came up with this integrated biomarker response, which was elevated whenever oysters were exposed to pathogens or pH, and it was further elevated when they were exposed to both. So, like I mentioned before, I'm doing a lot of work in BC, and I was asked to kind of share some of that work to spur some conversation about Alaskan work in oysters. In BC, the hatcheries, there are not very many of them. A lot of the seed comes from either Washington and Oregon or other places in the world. There's also a little bit of wild seed collection too, but the hatcheries that do exist have to buffer because they've observed the same problems that Oregon and Washington hatcheries observe. If they don't, the larvae don't develop. There is a very important program about selective breeding to try to target acidification resistance. That work is led by Dr. Tim Green at Vancouver Island University. And they've recently published some results from that work. Here you're seeing development of larvae at 24 hours on lines that were resistant to OA and lines that were not resistant to OA. The important thing to see here is that this last column is the larvae that are bred to be more resistant to acidification. And when placed on saturation state that is corrosive, only half of them developed, and even of the, which is the color bar right here, and even of the ones that develop a tiny fraction, which is the blue one here, actually develop normally. So this is tremendous amount of work, really hard, and they're making progress, but we're not quite there yet in getting an ocean acidification resistant strain. There have been, for quite some time now, events of summer massive mortality, particularly around the Bain Sound location, which is where about half of the entire region of oyster production is located. Those summer mortalities seem to be associated with Vibrio infections, but they also seem to be triggered by environmental thresholds. So warming, heat wave exposure prior to the infection, but we're starting to think that there might be a component of corrosive water as well. Like you recall from before, when oysters are exposed to corrosive conditions and Vibrio infection, their immune response is severely depressed. There was also, of course, a lot of massive mortalities, particularly on mussels, but also on oysters associated with last year's summer heat dome. But we don't necessarily think that those were related in any way to acidification, though there are concerns right now about loss of genetic diversity due to those massive mortalities, particularly in some locations, that could maybe lead to reduced resiliency for those populations to ocean acidification in the future. What I'm doing right now, I know this is a busy slide and I'm gonna try to be really quick here. What I'm doing right now in my work in BC is trying to understand how actual natural conditions, which are really variable, both in temperature and in CO2, will affect oysters in BC, particular oyster populations in BC, but also how the exposure to their parents may actually inform the response of the larval progeny or the larval offspring. For that, we went to Pendrell Sound in collaboration with Wiley Evans' team, and they had deployed there some sensors, so they measured pH over quite a bit of time. Pendrell Sound is an important place for us because it's one of the only places where there's wild natural recruitment of Pacific oyster, which, remember, is a non-native species and rarely breeds on its own in BC and definitely in Alaska because waters are too cold. So Pendrell Sound is an exception to that. We went there, we got data, and then we modeled what that data might look like into the future. And we brought oysters back into the lab and we tried to recreate that variability in the future in the lab. We're right now analyzing the data, but we can already see a pretty strong pattern in the adults. So exposure to corrosive or more corrosive conditions does lead to an increase in reproduction or acceleration in reproduction. They get ripe faster, basically, and they produce bigger but fewer eggs. That's potentially problematic if this keeps happening and turns the clock a little bit earlier in the season, because there's a careful need for these larvae in tendril sound to be released at the right time where there's enough food in the water. But there's also, we're still in the preliminary stages here, but it doesn't seem to be a very strong beneficial effect of parents passing any sort of information to their offspring to make them more resilient. This has been shown for other Pacific oyster populations in other parts of the world, but BC doesn't seem to be one of those. Alternatively, though, we're not seeing the same detrimental effects in the adults as other people in other parts of the world have seen. So there are definitely some differences in how BC populations are responding. And it's important to keep that in mind when we're trying to condensate data from multiple places of the world. But yeah, exposure of parents doesn't seem to be a great solution in this case. So
Iria,
just to wrap it up.
I'm going to interrupt you here. I know that your last slide is conclusions, and we're running quite a bit over at this point. So we're going to be able to share your slides, including the conclusions, to everyone. I know that there were some rich slides in there that everyone will be able to take a closer look at, and we'll be able to allow some back and forth in your breakout session. So I'm going to take this moment to say thank you so much for a rich discussion and some really positive feedback in the chat. And I'm going to make some room for our other two presenters still here before, hopefully we can still round it out in the next, maybe before the top of the hour. So thank you so much, Iria, for that presentation. That was terrific. Our next presenter up is Dr. Amanda Kelley, who is with the College of Fisheries and Ocean Sciences at the University of Alaska Fairbanks. And Dr. Kelley's research program encompasses understanding organism to environment interactions in the context of ocean change. Dr. Kelley is keenly interested in characterizing environmental sensitivities and thresholds that exist for high latitude coastal ecosystems in response to the changing environment. The focus of this work is twofold. First, her research is concerned with understanding the drivers of nearshore carbonate chemistry variability in high latitudes, using in-place techniques to measure factors associated with ocean change, including pH and temperature, et cetera. These data then are also used to frame laboratory experiments aimed at determining thresholds of coastal species to ocean change. Since arriving at UAF, Dr. Kelley's focus in part has been to study the impacts of future climate change on species that are important to Alaska Native communities as subsistence resources, particularly in the context of food and cultural security. So we're very excited to welcome Dr. Amanda Kelley. Amanda, are you in a position to share your own slides?
Yeah, just give me a second here.
Great.
All right.
Thank you.
Can everyone see my slides? Awesome. Yeah. Thank you so much. Thanks for the introduction, Molly. I would like to start off briefly. My name is Amanda Kelley. I'm an assistant professor at the University of Alaska Fairbanks. The title for this section of today's workshop talk is Nearshore Conditions and Responses of Subsistence Species to OA. I would also like to take a moment to do a land acknowledgement because this work has taken place all over the state. So the University of Alaska students, faculty, and staff reside, work, and study on the traditional and contemporary unceded homelands of the Haida, Yupik, Cupik, Supiaq, Alutiiq, Unganuk, Aleut, and Inupiat people. We recognize and honor their past, present, and future stewardship of this land, animals, water, and relations. We further recognize and seek to rectify the erasure of Indigenous people and the continuing legacy of colonialism in our university and scientific communities. All right, so I'm going to take it where Iria left off here. The real focus of my talk is kind of characterizing what those nearshore conditions are and framing what commercial species experience, but then again going into subsistence species. So here is a map of all of the mariculture operations across the Gulf of Alaska. And from our speakers last week, you learned about the factors that control carbonate chemistry offshore in these larger ocean basins. And they differ pretty dramatically from the conditions and the factors that control these conditions in the near shore. So for instance, nearshore carbonate chemistry, or pH, and other carbonate chemistry parameters are really influenced by the fact we see a greater range or amplitude in salinity, which is driven by freshwater inputs from glacial melt and precipitation, greater ranges of temperature, tidal fluxes, air-sea flux, and biological factors. And this one is particularly important given that the Gulf of Alaska is extremely productive and can be nutrient rich, driving really high biomass in some of these nearshore areas. So factors that control biological factors such as respiration and photosynthesis can really drive diel or daily fluctuations in some of these parameters. And so this map is really meant to just kind of highlight the different areas where aquaculture or mariculture exist here. And I wanted to give a brief overview of two places where we have nearshore carbonate chemistry data to kind of frame how, even though these two areas, one at Kachemak Bay and the other at Juneau, are in the near shore across the Gulf of Alaska and arguably receive the same source water across the Gulf in terms of the Alaska coastal current, other factors, including the amount of freshwater input, mixing, and biological activity, can really alter the chemistry in these conditions. And so first off, I just want to start off with sites in Kachemak Bay. So Jackaloff Bay is an area of active mariculture. There's an oyster mariculture operation that's operating right there. Again, Jackaloff Bay actually receives very little freshwater input. And so, but also it is an area of really high biomass, meaning there's lots of animals that live on the bottom as well in the summertime. When you go scuba diving there, there's like 3 feet of saccharina, which is a benthic prostrate kelp that just covers the bottom in addition to phytoplankton. And so you can see here generally, a lot of the threshold conditions that we see set for oysters and other experimental approaches often range about 7.6. And for the Gulf of Alaska, at least here in K Bay, the mean average pH is roughly 8, although we see pretty dramatic shifts when we get into summer and fall. So that's sort of our baseline for our experiments. And so when we think about what future conditions look like, it's roughly about 7.6 pH units. And you can see here on my graph that that's right about here. And so at least in places like Jackaloff Bay, we don't see these corrosive conditions happen basically any time of year. So if you're growing oysters in Kachemak Bay, although we do see brief periods of lower pH conditions, it's not necessarily that they're corrosive. And this is also verified by this plot here, where on the right y-axis, we have aragonite saturation state. And as Iria mentioned, periods that run below 1.5, here we do see that happen in wintertime. Generally speaking, for adult oysters, we stay well above this aragonite saturation state of 1. So not as corrosive as other places. And so I wouldn't necessarily say that Kachemak Bay is a nearshore hotspot. But then we can look at some work done by Wiley Evans and Evans et al. 2022. And this was the ferry data where they took surface carbonate chemistry measurements. And so what I want everyone to focus on here is this map. I'll first kind of frame this. This is Ketchikan is roughly 55.3 degrees north latitude, and then Juneau is roughly 58. And so this box here kind of represents the aragonite saturation state conditions throughout the year. We have on the x-axis here periods through winter, spring, into summer. And what you can see basically is anything below this 1 threshold are periods of time where we are sitting at below that threshold of 1 in terms of the aragonite saturation state. So compared to a place like Kachemak Bay, we see extended periods of seawater corrosivity inside Southeast Alaska. And so really the point I'm trying to make here is that conditions across the Gulf are not similar. They're highly dynamic, and certainly different factors control them. All right, so I don't have a ton of time, but I'm going to try and zoom through this here. So one of the things that I like to point out is that when we think about climate change impacts to invertebrate species that are important for subsistence resources, we don't know a lot about their basic life history. So for example, we did this work at the Alutiiq Pride Marine Institute with Jeff. At this point, we are interested in understanding how the larval period, which is typically the period in bivalves that is very susceptible to OA, would respond to these corrosive seawater conditions. And so we knew that the razor clam was in decline across populations, different populations in Alaska, and low recruitment was thought to be one of the factors. And so again, we just didn't know a lot of basic facts about their life history and biology. So that was one of the things that we needed to do to really understand how they might respond to climate change. And more broadly, understanding these basic biological processes that occur in their early and midlife history stages are really important because, you know, it's hard to know whether species will be susceptible or resilient because we don't know a lot of factors about aspects of their life history. So briefly, this experiment was conducted over 28 days, and for us, this was at the Shellfish Hatchery, Marine Institute, excuse me. The ambient was roughly 350 microatmospheres of PCO2 and 8 of pH. In our high or future conditions, we used an offset of 0.3, which is the IPCC business as usual model for the year 2100. But then we also did a variable treatment because what we saw in Kachemak Bay is that there was a daily diel variability that was a product of photosynthesis and respiration. So we wanted to include some degree of natural variability in this experiment. So this was our third treatment. It fluctuated between what we would expect to be ambient in the future, and then during the day it would rise up to 8 and then go back down at night. All right, we're going to jump right to the results. These are SEM micrographs of the shell development of razor clams, and what you'll notice, as Iria pointed out with the oyster, that they really start developing what looks like a pretty finished or well-polished shell by 16 hours. Conversely, the razor clam, as we ended up learning, has an alternative form of shell development. They have this, what we found to be a concretion. And so this is day 7, day 14, day 21, and 28. And what you'll notice is that this is just really an amalgamation of shell, and then they become more mature and develop this smoother shell. And what we found was that using Raman spectroscopy, which basically measures the level of organization of the crystal, is that they first develop amorphous calcium carbonate, which is just kind of a conglomeration of ions. But as they move to day 28, they actually produce vaterite, which we found out, this is a really cool discovery, it was a very biologically rare polymorph of calcium carbonate and is only in existence in a handful of different species on planet Earth. So this was really neat that we were able to find that. So when we think about how the development, understanding the basic biology of the species, how that might be important in terms of OA and other climate change factors, what we know is that here, ACC, or amorphous calcium carbonate, is the most soluble form of calcium carbonate. So we can think about using that Alka-Seltzer, that old Alka-Seltzer commercial, when you drop the Alka-Seltzer in water and it starts to actively dissolve. Both ACC and vaterite are actually the two most soluble forms of calcium carbonate. So it was really an interesting discovery that the larval shells were made of this polymorphic calcium carbonate. Potentially, you know, for us, maybe creating a potential reason why we might be seeing a decline in the population. But then we asked ourselves, well, what do the adults look like? And so we again used Raman spectroscopy and found that, in fact, they also precipitate vaterite as adults in their shell. So this particular bivalve is really typically more sensitive than other bivalves to ocean acidification. This production of concretion and then their more mineralized shell is really a product of the fact that this particular genus of bivalves is more basal, meaning it's some aspects of its biology are more closely related to gastropods, and gastropods, meaning snails, actually use this concretion as a shell developmental strategy. So again, identifying these basic biological principles is important. We also were able to secondarily identify the concretion because the shell in its earlier stages is made up as a whole amalgamation of different ions. Whereas when we get to day 28, it's mostly calcium, indicating calcium carbonate. The other thing we want to point out here is the fact, so this is looking at the 0.1 and the 0.2. 0.2 is this leading edge. So this was able to kind of tell us how the treatment was affecting shell development through time. What you'll notice here with these red boxes, this purple here is the calcium component of the measurements we were making. And what we found was that the high treatment on day 21 and then the variable and high treatment on day 28 actually accelerated their shell development, meaning they incorporated and moved to a more calcium carbonate vaterite based shell than the ambient treatment did. So that was one of the interesting results from our study. So the next species I'm going to talk about is the Darky. As I mentioned, much of our work is concerned with primary characterization, of basic biology of the species. The Darky is a really important food source for coastal Alaska Native communities. It ranges from California all the way across to Russia. It's an important intertidal grazer and creates a trophic link within the food web. And so, again, certain populations of the species were in decline in larger Cook Inlet, the Kenai Peninsula. So we were interested in maybe trying to characterize what their response would be to future climate change conditions. And so we did a multi-stressor experiment looking at increased temperature and decreased pH across 28 days. And the results for this study were a bit different. We basically found that grazing rate was significantly different, but only as a result of temperature, not acidification. And furthermore, when we were looking at the relative amount of dissolution, so here's a cool SEM image, we were actually able to determine that this laminar shell dissolution was not affected by OK conditions. And so we found no impact of elevated temperature or PCO2 on the respiration rate, the shell composition, or shell dissolution. The only thing we really found was that was affected was temperature through significant increased grazing. And I'm going to try and finish this really quickly here. Another thing we've been interested in looking at is we know what the overlying water column is doing, but a lot of the organisms we're interested in looking at, particularly for subsistence species, live inside the sediment. They're macrofauna, macroinfaunal species. And so we were interested in characterizing two different sites in Kachemak Bay. So here's larger Kachemak Bay, Homer's over here. The Kissisna Bay Lab is here. So one of our sites was at a rocky beach here where the beach is composed of shale and is quite porous. Whereas here in McDonald's Spit, it is a tidal mud flat. And so in places like Kasutsina Bay, we do have cockles and some little net clams that are sort of epibenthic or live on the surface, where in McDonald's Spit, we see more infaunal species such as butter clams. And this was done in July 2020. And what we found in Kasutsina Bay, and it's not super surprising that the pH is relatively low, 7.5 to 7.8, but it makes sense relative to what we see in the overlying water because of respiration that's occurring down here. But interestingly enough, McDonald's Spit is less porous, so there's less of a flushing rate. We actually saw quite a bit more acidic conditions here with pHs reaching 6.2. So again, I just wanted to point out here that the distance here between Kasutsina Bay and McDonald's Spit is about 1.5 miles, but given the specific conditions of each one of these beaches, it can really affect the pore water conditions there. So I'm going to wrap this up really quickly. The take home here is that surface waters are corrosive in Southeast Alaska. Pore waters in some sites in Kachemak Bay are corrosive. K-Bay is actually, the overlying water column tends to be less corrosive. Razor clams are likely susceptible to ocean acidification due to their alternative shell developmental strategy because they have amorphous calcium carbonate and baterite as opposed to calcite as their main shell mineralization. And then finally, Darkies, or the black chitin, appear to be pretty resilient. We found no effects of pH and only one modest effect of temperature on grazing. And with that, I'd like to say thanks for everyone for listening.
Dr. Kelley, terrific. That was wonderful. Thank you. And I'll just put a little plug in that for those of you who want to start thinking about where you'll go for your breakouts, we're going to give everyone the option of choosing. And Amanda will be in one of the groups, and that will focus on subsistence species that she mentioned already. And Iria will be in the other group focused on Pacific oysters that she was covering in her presentation. So you can be thinking about which of those two groups you might go into. Jeff Hetrick is our next speaker, and he'll be joining Amanda in one of the breakouts. He's the director of the Alutiiq Pride Shellfish Hatchery in Seward. He has 30 years of experience in the Alaska aquaculture industry and formerly operated a shellfish farm in Prince William Sound. He's been the director of Alutiiq Pride since 2002. They started looking at ocean acidification during the shellfish hatchery crisis that affected the oyster industry on the West Coast. And it was in response to concerns that were being expressed by the hatchery managers and operators, which others have alluded to here already. They were asked by researchers to maintain a monitoring site and agreed to do that, and have been utilizing a shellfish garden and sanctuary concept to help bring back some of the localized shellfish populations near native villages in the Chugach region. So we'll be hearing, I believe, some more about that from Jeff. So thank you, Jeff, for joining us. And you ready to share your slides?
Yeah, good afternoon, everybody. Thanks for Iria and Amanda, colleagues in this whole OA thing. So like Molly said, we got into the ocean acidification studies when the shellfish hatcheries in the Pacific Northwest had some major crashes starting in the late 2000s. And we were asked to maintain one of the original continuous monitoring devices, and eventually got what we call a percolator. What you're going to hear from me this afternoon is a little bit different. We're sort of a vector between the science, the pure scientists, and the communities, trying to translate scientific data into things that are meaningful for people who live in coastal communities, native communities, and shellfish farmers around the state. And we're fortunate that we have a lot of resources here at the Alutiiq Pride Marine Institute that we can not only monitor ocean acidification, but help other communities with their samples, and then do some research on what the effect of OA is. Dr. Kelley did mention about the life histories. That's one of the major things that we do. We work with a lot of species that people really don't focus on. You know, for instance, when a little net clam spawns and what their fecundity is and how their larval behave and what their growth rates are, what their mortality is. We're sort of a salmon, halibut, crab state, and not a lot of people pay that much attention to the importance of our benthic communities, and especially on our beaches in front of our communities. So the Alutiiq Pride Marine Institute is operated by the Chugach Regional Resources Commission. It's an umbrella organization that does far more than just Alutiiq Pride Marine Institute. We have wetlands and we have subsistence, food security. We deal with fishing game and federal subsistence issues, and have a pretty large staff that does more than just Alutiiq Pride Marine Institute. But they all come together with the same intent and desire to protect the natural resources, understand them throughout the Chugach region and also throughout Alaska. The communities we serve in the Prince William Sound are Valdez, Titlik, and Eyak on the eastern side, Chiniga in the south part of the Prince William Sound, Katuchek here in Seward, and then we have Port Graham and Nanwalek, and a really good partner in Seldovia, which is near Kasitsna, what Dr. Kelley talked about. This is what's called a percolator. This was the second or third generation of ocean monitoring equipment that kind of came as an artifact of the crisis in the shellfish hatcheries in the Pacific Northwest. It's a device that it's operated by our lab manager, Jacqueline Ramsey. It takes a fair amount of time, but it's really kind of an interesting device in that it continuously monitors Resurrection Bay, and it's tied into a network where people can actually monitor what's happening here in Seward continuously. But it also has the capacity to do discrete samples. And so what that means is that we can take it offline, and from a few manipulations, we can sample or take readings from samples that come in from our communities. So what's really been interesting is that when we first started looking at the OA levels in Resurrection Bay here in Seward, we found some levels that were kind of alarming to research scientists. But it wasn't really totally a surprise. Resurrection Bay has a lot of things that cause OA. It's got deep fjords, a fair amount of freshwater influence, glacier. So it wasn't totally surprised that we had some levels that were of concern. But it caused us to ask the question, is that just here, or is that region wide? And then we took advantage of a program we have where we have environmental coordinators in all of our communities. And as part of their duties that exceed obviously marine sampling, we train them to collect samples for us. And once a week, they would go to their dock, collect a bottle sample, and preserve that sample. And after a certain number were collected, they would mail them to the facility. And Jacqueline and her lab would process those samples. And what we found is that, the levels were obviously different from one place to another, but during certain times of the year, under certain circumstances, we had OA levels that were of concern. So we've been doing this now for 10 years. We've expanded our sampling base to go out to Western Alaska. We go as far as Ukigagvik North. We have King Cove. We've got through Ayus, we have samples in Nome and Kotzebue. And so we continuously process these samples to try to get a long-term data set that helps people better understand ocean acidification. So what I'm going to talk about really is sort of what we're going to do about this. We can monitor and we can model, and those are all really valid exercises to do. But what exactly is it that we might be able to do to deal with ocean acidification? And so what you see in this photograph is what we call a PCO2Go. It's the latest device by Dr. Hales. That's a field instrument. And it's being funded by actually the government of Sweden through the Ocean Foundation. And our responsibility is to do the beta testing, and we're going to get the second version here pretty soon. And this device is developed for hatchery operators, and perhaps there's many other uses as well. But places that don't have access to a percolator, don't have access to some of the fine marine sampling program that many of us have at our fingertips. And this device is simply made to, if you have alkalinity that's fairly stable, it doesn't wander too far, and you get the salinity and temperature, and this device measures PCO2, then you can calculate your uragonite saturation levels, and you can also calculate TCO2. So you take this device, it's pretty simple, and you put it into your tank, and then through a thing called the manipulator, you can plug it into your laptop, and it will tell you how to dose your tank. So if you're having problems with your chemistry, for the most part, you can add soda ash or lime to your tank, which adjusts the pH and therefore makes your levels what they need to be. It's really, really kind of a neat device. And there's two parts to it. One is the device that does the measurement. But the second thing that's really been enlightening for us here at APMI is that you can put in different numbers into the spreadsheet on the right, and it can do these calculations for you. It's a lot of numbers, but the reality of it is it just kind of gives you a sense of how, just by moving the alkalinity a little bit or moving the PCO2 value a little bit, how much it changes and how really dynamic the study of ocean acidification is. And so being a part of this process for over 10 years and listening to people like Dr. Evans and listening to Dr. Hales and these folks, you know, and trying to grasp bits and pieces of what they're telling you to understand the whole dynamics of ocean acidification and the carbon chemistry, the PCO to go and the manipulator have been really valuable for, at least for myself and some of our staff, to get a real good grasp of the dynamics of ocean chemistry. So as Dr. Kelley mentioned, she does some pore sampling in Consistent Bay. We actually do some of our own in Seldovia, but I see this device being really valuable for that. Our second version that we're going to also place in Seldovia and probably Port Graham. And then we can do some more calibration of the PCO to go with the percolator here at the hatchery and just further develop the equipment. So one of the things that we did this year was we just wanted to use the PCO2 as a tool in the hatchery. And we did a real simple experiment. It certainly wasn't a scientific, didn't have a lot of controls. What we did was just try to use the manipulator to dose our tanks at different levels and see what the effect was on some eyed oyster larvae. And you can see just a simple graph, it's pretty dramatic, that the survival goes dramatically down at 1.2 and our ambient is 1.7. I need to let folks know here that it's not uncommon for us to have levels of 1.3. In fact, that's probably the most common level we observe at the hatchery, especially during the winter months. So we've been raising shellfish at this lower level for 8 years now. It's not until we have these tools and this capacity that we can actually dose our tanks and hopefully increase their survivals. Coming up this spring, we're going to be doing, we'll be dosing all of our tanks, getting them to the 1.7 or above 2 levels. And of course, we'll do some ambient temperatures, or excuse me, ambient levels, and see what the difference is. So it's kind of like applying the pure science to the applied science. So what I just told you about was how we adapt for ocean acidification in the hatchery itself. But what are we going to do for our communities? So we do a lot of research on butter clams. Dr. Kelley didn't mention some other things that we've done as a team, but butter clams and cockles and little neck clams. But one of the things that we found with all of our work in the field is that there's a species of clam called the soft shell clam, and we call it a non-traditional food source, but it's classified as an intentional and unintentional introduction into Alaska. But as we go out to these beaches and we're enhancing or working on little neck clams and butter clams, we seem to find that these soft shell clams are doing exceptionally well. And there's been discussion that perhaps they too have a different way of putting on shell, such as Dr. Kelley mentioned with razor clams. But for whatever reason, not just OA, but increasing temperatures and high energy events and all the things that are sort of the menu for why there are not a lot of clams anymore, the soft shell clam seems to be doing pretty well. So we started raising them in a hatchery a couple of years ago, and they're doing exceptionally well. They grow really fast. We started a few outplanting experiments to find out, you know, how fast do they grow, find out a lot more about their life history specific to Alaska. But it might be that in a few years, maybe as short as 10 years from now, that we'll need to tell the folks in our communities that one of the clams they can begin to look at harvesting and consuming is something that's really not part of their natural diet, and that would be soft shell clams. So in the future, what we're going to do here is continue this kind of work, you know, applying pure science to what we call applied science and keep on working with our communities to try to get a better understanding of ocean acidification. And that's all I had this afternoon.
Thanks, Jeff. That was terrific. Thanks to all of our speakers, Amanda and Iria also. That was a great setup. So let me, since we use the majority of the time for presentations, let me suggest that we take follow-up questions into the breakouts. Since Jeff, Amanda, and Iria have all agreed to join us in breakout sessions, we can use some of the beginning of those sessions to cover any follow-up questions that might be beneficial. So we have an option of one of two different breakout sessions that we'll present to you. You'll be able to make your choice. You can join one or the other. One will be focused on the subsistence species that Amanda and Jeff just focused on in particular, and the other one will be focused on Pacific oysters. Each group will have experts and a facilitator and note taker. We're also going to give the group, for those of you who are in the last session, you will already have been introduced to a mural whiteboard, but we're going to use another tool for another mural tool for this one to allow people to participate, not just verbally, but also in writing. So we'll introduce those to you in the breakout sessions. So be on alert. What we'll do is we'll move into the breakout and then we'll provide a link for you to join that mural and communicate in a variety of ways through a facilitated conversation in your breakout. So let me turn to Darcy and ask Darcy to share with you a choice to go into one of two different breakouts. And we'll automatically bring the group back together again at about 10 minutes till. We're going to cut it a little bit shorter this time since to give us as much time as possible in the breakouts, but you'll be automatically brought back into the plenary at about 10 till, and we'll share some of the discussion that took place with everybody together here back in the plenary before closing out at the top of the hour. So you should see on your screen a window that shows, if you scroll to the bottom of the breakout room window, you should be able to select to join either Pacific oysters or subsistence species, and you just click join and you'll be switched over into that breakout room. And if you don't see that window, then you may have to click on more at the bottom right of your screen, and it should be able to show you options for joining a breakout room. All right, and we'll see everybody back at about 10 till.
I can't seem to find how to do that.